Hello, and a very warm welcome to everyone joining us for today's Select Science webinar entitled Electrical Field Stimulation for the Functional Maturation of Human-Induced Pluripotent Stem Cell-Derived Cardiomyocytes. My name is Matt McArdle, and I'll be moderating today's presentation. I'm delighted to introduce today's expert speakers, Dr. Xiaoyu Zhang, Principal Scientist at ACEA Biosciences, and Dr. TK Feaster, Product Manager at Fujifilm Cellular Dynamics. In the webinar today, we'll be talking about principle and protocol for electrical pacing of cardiomyocytes, measurable characteristics of matured cardiomyocytes, a new instrument that allows high throughput maturation, and implications on the future of cardiac research, including workflow models. After the webinar presentation, we'll move on to our question and answer session. So please feel free to ask any questions for the Q&A session at any time during the webinar, and you can submit your questions at the left of your screen. Without further delay, I'm delighted to hand over to Dr. Xiaoyu and Dr. Feaster. I want to thank them both for presenting to us today.
Thank you. So today I will focus on two topics, electrical stimulation and human iPSC-derived cardiomyocytes. So first, I will discuss the application of electrical pacing for human iPSC-derived cardiomyocyte study, and then I will talk about immaturity of these cells and its limitations. At the end, I will show you how to use long-term electrical pacing to enhance maturation status of the cells using our exogenous RTCA pacer. So as we know, each of our heartbeats is triggered by electrical impulse, which initiated from sinoatrial node and propagates throughout the entire heart. So even during the development of fetus, the heart beating activity can be detected as early as 5 weeks of gestation and continuously change throughout the entire gestational period. So all these facts indicate that the electrical stimulation is a natural process of our heart. It may also play a very critical role in our heart development. So now I would like to discuss what we can do with this natural process electrical pacing for application of human iPSC-derived cardiomyocytes. So far, the short-term electrical pacing has been used to study rate-dependent changes in regularization duration. In addition, correcting beating rate across different cardiomyocyte lines would help scientists to get better phenotypical profiles of the cells of their interest. So in this study, we are trying to evaluate hypertrophy cardiomyocytes by comparing to its isogenic control. So as we can see from these top two figures, the beating waveforms here, the disease line has much slower beating rate than the control line. In order to correct the variation of the beating rate, we actually applied electrical pacing to control beating rate at 0.5 Hz in both lines. The field potential duration and the force of contraction were then evaluated when two cell lines contract at the same rate. So the hypertrophy cardiomyocytes clearly showed the stronger contraction force and the longer field potential duration than the healthy line at the control beating rate. So in addition, our most recent data indicates that the long-term electrical pacing can improve maturation status of the human iPSC-derived cardiomyocytes. So my following slides will demonstrate that functional maturation of these cells significantly increases the predictivity for the drug-induced cardiac toxicity, the effects of inotropic compounds, as well as revealing disease phenotype. So immaturity of human iPSC-derived cardiomyocytes is a long-standing problem in this field. So when comparing to the adult cardiomyocytes, human iPSC-derived cardiomyocytes still show fetal-like phenotypes, such as irregular cell morphology and organized sarcomere structure, higher resting membrane potential, immature calcium handling machinery, smaller force of contraction. In addition, human iPSC-derived cardiomyocytes possess negative force frequency relationship, which is completely opposite to adult cardiomyocytes. The negative force frequency relationship of human iPSC-derived cardiomyocytes has been reported by numerous publications using different technology, such as video edge detection as shown in this left-hand figure. So this figure here shows that when the beating rate increased, the contraction reduced. In our hand, the impedance data obtained from exogenous RTC cardio ECR system also demonstrated that the increase in the beating rate controlled by electrical pacing reduced the beating amplitude, indicating the negative force frequency relationship as well. So due to the immaturity, human iPSC-derived cardiomyocytes still cannot be fully utilized, especially for assessment of inotropic compounds. So in this paper, a panel of contractile modulators, which are inotropic compounds, were tested in human iPSC-derived cardiomyocytes. The contractile force was evaluated by the extent of shortening of sarcomere length observed on ion optics instrument, which is an optic-based system. So their data showed that human iPSC-derived cardiomyocytes had good predictivity for negative inotropic compounds. However, when these cells were treated with positive inotropic compounds, they displayed either no effects or even reduction in the force of contraction, suggesting these cells is not a proper cell model for this application. So, in addition, these cells showed limited prediction for drug-induced arrhythmia in comprehensive in vitro pro-arrhythmia assay (CPA) study. So during CPA phase 2 study, 28 compounds were categorized into low, intermediate, and high arrhythmia risk based on their clinical outcomes. The compounds were then evaluated in 10 individual test sites. The results here showed that human iPSC-derived cardiomyocytes had good predictivity for high-risk compounds. However, they showed a limitation for predicting low-risk compounds. So as you can see here, several sites show that some low-risk compounds induced arrhythmia, which is not consistent with clinical results. Even for cardiac disease model, study immaturity of the cells may also affect assessment of disease phenotype. So in this study, we were trying to identify the disease phenotypes of cardiomyocytes derived from the patient with dilated cardiomyopathy. We treated disease and isogenic control lines with isoproterenol, which is a positive inotropic compound. It seems that there were no difference between these two lines, as you can see from this left figure. The right bar here is the data collected from disease line, and the green bar is the data from the healthy line. So both lines showed reduction in beating amplitude. So after we improved maturation status of these cells using long-term pacing, the disease line still showed no effect or reduction in beating amplitude. However, the matured healthy line started to show increase in beating amplitude, which reflects the increase in the force of contraction. This result suggests that the defective contractile mechanisms in dilated cardiomyopathy. So the data here also indicates that improved maturity could help to tease out the hidden disease phenotypes. So up to date, a number of natural engineering approaches attempting to enhance the structural and functional maturity of human iPSC-derived cardiomyocytes has been described in literatures, including co-culture, mechanical conditioning, use different extracellular matrix with different degrees of stiffness, and electrical pacing. So in our lab, we have developed a maturation protocol using the electrical pacing function provided by cardio-ECR system. So this is the Excelligence RTC cardio-ECR system. It has 4 components, as you see from this picture. The laptop is the control unit for scheduling and performing experimental steps. And the station, which is the plate reader, is permanently placed in the incubator. The analyzer is used for data transferring from the station to the laptop. So, and this is the cardio-ECR plate, which has 2 separate sets of electrodes. The impedance electrodes are used for the impedance recording, and the field potential electrodes are used for the measurement of field potential signal. In addition, the impedance and the field potential signals are measured simultaneously. Due to the fast data acquisition rate of cardio-ECR system, the temporal changes in attachment and morphology of the cells during each cell beating can be captured and quantified by impedance readout via impedance electrodes. So in summary, cardio-ECR system can measure cell contraction and the cell variability via impedance readout, and the amplitude of the impedance waveforms indirectly reflect the force of contraction. And like other MEA systems, the field potential signal collected by field potential electrodes is used to evaluate integrated ion channel activities. So besides simultaneous measurement of these impedance and field potential readout signals, the system also provides a built-in electrical pacing function via these impedance electrodes. So due to the large coverage of impedance electrodes in each well, it only requires much lower power to pace the cells compared to other commercially available pacing apparatus. The lower power pacing setting ensures the cells won't be impaired by long-term electrical pacing. And the diagram here shows how we use 2 weeks electrical pacing to improve maturation of cardiomyocytes. So basically, we continuously apply a direct and progressive electrical stimulation to the cell culture for 2 weeks. At the end of the pacing process, we evaluated maturation status of these cells. So first, we tested force frequency relationship. The black trace here shows the beating rate increase resulted in a decrease in beating amplitude, indicating negative force frequency relationship before long-term pacing. However, after 2 weeks pacing, the cells started to show positive relationship as shown in the red curve here. So higher beating rate led to bigger beating amplitude. And in the meantime, the cell contractile responses to positive inotropic compounds were improved. So here we only showed isoproternal treatment as one of examples. So only the orange bars here, which represent the data from long-term paced cells, showed significant increase after iso addition. However, the blue bars, which is the non-paced cells, still showed reduction in beating amplitude at a higher dose. In addition, our immunostaining showed that the long-term paced cells had better structure or organization of sarcomere than non-paced cells. We also compared gene expression levels between paced and non-paced cells. Even though not all tested genes improved after pacing, we did observe more than 4-fold increase in KCNG2 expression in paced cells. So this suggests that IK1 current, which is the critical current contributing to the resting membrane potential, may improve in paced cells. So taken together, these results suggest the maturity of human iPSC-derived cardiomyocytes is indeed improved by chronic electrical pacing. So due to the importance of maturity of human iPSC-derived cardiomyocytes, our recommended assay workflow is shown on this slide. So first, use long-term electrical pacing, which is considered as a natural engineering approach to improve maturation status of the cells. This maturity-enhanced cardiomyocytes will serve as a better cell model for drug discovery, especially for inotropic compound screening. So in this study, we tested a panel of contractile modulators, including positive and negative inotropic compounds with different mechanisms of action. All the tested positive inotropic compounds, such as bemabandin, a calcium sensitizer, milrinone, a phosphodiesterase inhibitor, captive myosin activator, showed profound increase in beating amplitude, reflecting increased force of contraction only in maturity-enhanced cardiomyocytes. In addition, we retested ranolazine in both regular non-paced cells and functional mature human iPSC-derived cardiomyocytes. As we know, ranolazine is a dual ion channel blocker for both ERK channel and late sodium current. So due to the compensation of late sodium current blockage, ranolazine only induced QT prolongation, but no arrhythmia observed in clinic. Therefore, it has been categorized as a low arrhythmia CEPA compound. So as we can see here, in regular human iPSC-derived cardiomyocytes, ranolazine caused arrhythmia demonstrated by contraction traces and field potential signals. However, the mature human iPSC-derived cardiomyocyte didn't show any sign of arrhythmic events. In addition, the improved maturity could bring up disease phenotypes as shown in my previous slide. So as I mentioned previously, we use 2 weeks pacing method to mature human iPSC-derived cardiomyocytes. In order to produce more mature cardiomyocytes in 2 weeks time frame and further increase assay throughput, we developed Excilogen's RTCA ePacer, which serves as a useful equipment to generate better cell model. So now I'm going to go through some key features of ePacer. So first, ePacer provides high throughput. The user can pace up to 6 plates of cardiomyocytes at the same time within 2 week time frame. Second, it has very flexible stimulus setting. If you have done pacing study before, you would know it could be very tedious and time-consuming when you start to pace a new cardiomyocyte line, and especially when you do it for 6 plates of cells. The automatic optimization protocol for pacing setting will help users to figure out the optimal pacing parameters. In addition, the ePacer can actually help you to pace the cells in different columns with different pacing settings as shown in this figure. In the meantime, the system has multiple pacing protocols. You can actually generate artificial arrhythmia using arrhythmia protocol provided by the software. So in order to quickly evaluate cell maturation status, ePacer software has built-in function to calculate force-frequency relationship during the pacing process. Once your cells start to show the positive relationship as shown in this chart, you can initiate compound addition. And due to the compatibility of the plate, the enhanced cells produced by ePacer can also be used on other platforms, such as FDSS system for calcium transient measurement and the high-content imaging system for immunostaining. And the data showed in this table demonstrates that ePacer has very high pacing efficiency and very good uniformity throughout the entire pacing process. Last but not least, the chronic pacing provided by the ePacer do not harm the cells. As you can see from this overall cell index, which reflects the cell health and viability, didn't show significant decrease after pacing, shown in this orange curve, compared to non-paced cells presented by the blue curve. In addition, the ER stress markers did not increase in paced cells. So where can we place the ePacer in your IC workflow? Based on our experience, we propose the workflow as shown here. First, seed the cells in our ePacer plates, let cell grow and generate a stable beating activity, and then assess the beating amplitude and beating rate relationship using ePacer built-in algorithm. So once the cell reaches to ideal maturation status, and you can put the cells on exogenous cardio ECR system for cell viability, contraction, and electrophysiology evaluation, or you can measure calcium transient using FDSS system, or check the protein localization and expression using high-content imaging system. The action potential of the cells can also be measured using voltage-sensitive dye on optic-based system. The plate reader can also be used for these cells. So in summary, the contractile response, sarcomere structure, and gene expression of human iPSC-derived cardiomyocytes are enhanced and pushed towards the right path to mature cardiomyocytes by long-term electrical pacing. And electrical pacing, in combination with other methods, may further improve maturity of human iPSC-derived cardiomyocytes. And thank you.
First, I would like to thank you all for attending this webinar and ASEA for giving us this opportunity to share some of our research. My name is TK Feaster, and I'm the product manager for the cardiovascular portfolio at Fujifilm Cellular Dynamics. In today's webinar, we'll be talking about using human-induced pluripotent stem cell-derived cardiomyocytes and really trying to understand the biology of these cells a little bit more and the effects of pacing to enhance some of their functionality. I'll start off by giving you some background on Fujifilm Cellular Dynamics and induced pluripotent stem cell technology. Then I'll introduce the ePacer, which is a system that ASEA developed to pace these cells. And I'll show how that affects their force-frequency relationship and the inotropic response. Then I'll talk about detection of some of the outlier compounds in the SIPA studies, and then I'll use a quick summary to try to bring it all back together. Fujifilm Cellular Dynamics is based on stem cell technology. The whole idea with this is that one can take cells from any patient or any donor. This can be a simple blood draw from peripheral blood cells or fibroblasts from a skin punch biopsy. These cells are expanded in culture and exposed to reprogramming factors to force them into this induced pluripotent stem cell state. At this time, these cells can be expanded, banked, or genetically engineered to introduce a mutation or correct a mutation, and they can also be differentiated to different cell types. And these terminally differentiated cells are being used in basic science, drug discovery, cell therapy, and toxicity testing. Fujifilm Cellular Dynamics has an array of off-the-shelf cryopreserved products. This includes our brain and nervous system portfolio, which has astrocytes, dopaminergic, GABAergic, glutamatergic, and motor neurons, and we have recently released our iCell microglia. We have an ocular portfolio that includes RPEs, a blood and immune portfolio, and a liver portfolio that includes hepatocytes and hepatoblasts. But for today's talk, we'll be focused on the heart and vascular section, predominantly the iCell-Cardiomyocytes. iCell-Cardiomyocytes are really our flagship product. These cells were launched in 2009, and they have intact excitation-contraction coupling. This is electrical changes at the membrane being translated to intracellular calcium handling changes through a mechanism known as calcium-induced calcium release. This calcium then interacts with the myofilament and results in mechanical beating or contraction. In the lower panel, I'm showing an excelligence readout showing how one can measure this in vitro in real time. These cells are currently being used in the drug development process, but they're really fit for purpose, and it's important to understand the strengths and limitations of the system. This slide is a table from a review from Chuck Murray's lab in 2014, and it just really highlights some of the differences between an immature and a mature cardiomyocyte. For today's talk, we're going to be focused on the response to beta-adrenergic stimulation. It's well known that immature cardiomyocytes have an intact chronotropic response, however, they lack this inotropic response, while adult cardiomyocytes has the intact chronotropic response, however, they also have an inotropic response as a result of beta-adrenergic stimulation. There are many ways to try to bring out these functional benefits in an immature cardiomyocyte. One way to do that is through electrical pacing. In an effort to support this, ASEA has recently released the ePacer system. This system allows electrical stimulation of the cardiomyocytes in the assay plate. This is a great benefit because this means one does not have to dissociate the cells and plate them or replate them in order to do their experiment. You can pace the cells and run your assay in the same plate. This enables real-time monitoring to allow you to optimize your signal, and it has a built-in force frequency test to allow you to make sure your cells are ready to assay. The system also has built-in impedance and MEA signal for contraction and electrical activity, and there's a negligible cell loss as a result of the pacing. The system also has multiple ports to allow you to pace the cells while not having to tie up the system if you only have one port. So what we wanted to do was build on the experiments that ASEA has done in developing this pacing method. We wanted to extend this to different cell types. So here I'm showing the experimental design, and we tested 2 different cardiomyocytes from 2 different donors made with 2 different methods, the iCell-cardiomyocytes squared and the iCell-cardiomyocytes from donor 11713. These cells were thawed and plated in the E-plate according to our application protocol. 4 hours later, the medium was changed from plating medium to maintenance medium. And then on day 7, we began the pacing protocol, pacing the cells at 1 hertz for 5 days. This increased to 1.5 hertz at day 12 and 2 hertz at day 17. At day 22, pacing is stopped and the force frequency test is run. After the positive force frequency response, we applied the compounds. This was for a 30-minute incubation, and then did the experiment under spontaneous conditions. So the cells were paced to enhance their functionality, and then assayed under spontaneous beating. This is an example of what the waveforms look like on the ePacer. On the left-hand side, I'm showing the unpaced control, so these are cells that were never paced. And on the right-hand side, I'm showing paced cells at 3 different time points, day 12, day 17, and day 22. And the paced cells I'm showing at each frequency 1, 1.5, and 2 hertz. You can see that the paced cells are responding to the pacing. You can see that there's an increase with beating with every stimulus. And you can also see that the waveform of the paced cells is identical to the waveform of the unpaced cells. This makes data analysis very easy. After 22 days during pacing, we will then do the force frequency test. On the top, I'm showing the iCell-cardiomyocytes 11713, and the bottom, the iCell-cardiomyocytes squared. On the y-axis, I'm showing the amplitude, and on the x-axis, I'm showing increasing frequencies. The red is the unpaced control, and blue is the pace. So you can see that as you increase the frequency, the unpaced control, shown in red, has a flat or negative relationship. However, after the cells have been paced, this relationship becomes positive, and this is that positive force frequency response that you would see in a typical mature cardiomyocyte. The response is the same for the iCell-cardiomyocyte squared. You can see the unpaced control has a flat relationship. However, in blue, after you pace the cells, there's this increase in the force frequency relationship. One thing we noticed was that there was a reduction in the amplitude after pacing the cells. So we wanted to look a little bit at the baseline properties and see if there were any changes. Here I'm showing a quantification of that, where we looked at the iCell-cardiomyocytes 11713 on top, and we're showing the quantification for beat rate and then amplitude. I'm showing in green the pace cells have a reduced beat rate and a reduced amplitude, consistent with the previous slide. The iCell-cardiomyocytes squared, as shown on the bottom, actually have an increase in the beat rate, but they also have this same reduced amplitude. So it appears that there's going to be a reduction in amplitude that's induced by pacing. Here I'm showing the pharmacological response for isoproterenol and nifedipine. Isoproterenol is a beta-adrenergic agonist, and nifedipine is an L-type calcium channel blocker, which here is our control. I'm showing the iCell-Cardiomyocytes 11713, and on the left-hand side I'm showing the beat rate, and on the right-hand side I'm showing amplitude. You can see that as you increase the concentration of isoproterenol, there's a concentration-dependent increase in the beat rate. This is for both groups, the unpaced cardiomyocytes as well as the paced cardiomyocytes. So this is that classical chronotropic response that we know are intact. If you look at the amplitude, you can see that in black, the unpaced control, as you increase the concentration of isoproterenol, there's actually a concentration-dependent reduction in the amplitude. However, after pacing the cells for maturation, you begin to have a positive relationship displayed by this increased concentration of isoproterenol resulting in an increase in amplitude. The nifedipine control has a typical induced pluripotent stem cell cardiomyocyte response with an increase in beat rate for both groups and a reduction in amplitude for both groups. Next, we looked at the iCell cardiomyocyte squared, and we saw the same response. As you increase the concentration of isoproterenol, that beta-adrenergic agonist, there's an increase in the chronotropic response for the beat rate for both the paced and unpaced. However, the amplitude for the unpaced does not have an increase. So you have that, you're lacking that inotropic response. However, after you pace the cells, this becomes present. And the nifedipine control checks out. Next, we want to do an independent experiment to confirm what we saw with the impedance readout. Here I'm showing unpaced control on top and paced on bottom, and this is a calcium handling readout for vehicle and isoproterenol at 0.1 micromolar. You can see that when you expose the cells to isoproterenol, there's no increase in the calcium transient amplitude for the unpaced control. However, for the paced cells, you have the significant increase in the calcium transient amplitude, which is consistent with the impedance amplitude readout. This work was done using the cardio E-plates from the CardioView 96. These were paced in the E-plate and then moved directly to the calcium handling imager. One can also take these cells and move them to a high-content imaging system, as I'm showing on the right, staining of the cells for cardiac troponin T and DAPI. Now we're going to transition and talk about detection of some of the outlier compounds from the SIPA study. This paper was published in 2018 by Blanova et al. In Cell Reports. This is really a landmark paper that is from multiple groups across multiple contents with 28 compounds. These were all blinded with one protocol, and there was greater than 87% predictivity found. This is a very powerful study. However, it's not perfect. There were some compounds that were misclassified. For example, bepredil, ranolazine, metoprolol, and mexiletine. Bepredil is a high-risk compound that should result in arrhythmias. However, it does not reach the threshold. The low-risk compounds, ranolazine, metoprolol, and mexiletine, are actually approaching this threshold, so they could be misclassified. We wanted to take a look at these compounds in cardiomyocytes that had been paced to enhance their functionality and see if we were able to classify them correctly. Here I'm showing the result from that experiment. On top I have the ECR signal, which is the electrical signal. On the bottom I have the impedance signal, which is the contraction signal. I'm showing increasing concentrations of the compound. For low-risk compounds, ranolazine, mexiletine, and metoprolol, and bepredil. On the lower panel, I'm showing the SIPA concentration that was effective in the SIPA studies, as well as the Cmax for that compound. You can see that for vehicle, there's stable beating as shown in the representative ECR and impedance trace, and no irregularities shown in the table. For ranolazine, at 100 micromolar, the cells are quiescent. This is the concentration in SIPA that resulted in irregular activity. Mexiletine, at 10 micromolar, you can see on the table at 11 micromolar, the cells are quiescent. For metoprolol, at 100 micromolar, they're also quiescent. So you're seeing that there's no irregular activity with these compounds after pacing. And that's consistent with bepredil. The cells are quiescent at 100 micromolar. So it looks like after pacing the cells, one can correctly identify these low-risk compounds. However, the high-risk vector is still misclassified. So I've shown you data how you can establish a positive force-frequency relationship using the ePacer in human-induced pluripotent stem cell-derived cardiomyocytes. I've shown you how this leads to detection of positive inotropic compounds such as isoproterenol, and this can be multiplex with other readouts such as calcium handling. I've shown you how pacing the cells to enhance their functionality can allow one to identify some of the compounds that were outliers in the SIPA studies. And I've also shown you how you can pace the cells. It may alter their beat rate and their amplitude, but you can see this effect. With that, I believe we're going to take questions.
I'd like to thank both speakers for that interesting seminar, and I hope that everyone listening enjoyed the webinar and found it informative. We'll now move on to the last part of today's webinar, the question and answer session. We've got a few questions here. Our first question, I'm not sure which speaker would like to answer this, but could we talk about what age do you begin the electrical pacing of the iPSC-Cardiomyocytes?
Yes, this is TK. I'll take this one. The ICL-cardiomyocytes and the ICL-cardiomyocytes squared are approximately day 30 of differentiation when the cells are plated, and then 22 days later, the experiments are performed.
Well, thanks for that, TK. Our next question is, did these 2 weeks long-term pacing protocols stress the cells at the end?
Hi, this is Xiaoyu. I will take this question. So actually, one of the nice features of our ePacer is that it has built-in cell QC function via impedance readouts. So you can check the cell variability and the beating activity during long-term pacing process, which actually can help you evaluate cell health and the pacing capture efficiency in real time. So as I have showed in one of my slides, based on the results from multiple separate experiments, the overall cell index, which is a surrogate for cell variability measured along the pacing process, didn't show significant change compared to non-paced cells, which actually suggests the pacing provided via ePacer didn't affect cell variability. In addition, the protein expression of ER stress markers didn't increase compared to the non-paced cells, which suggests that no or minimum stress occurred during the pacing process.
Brilliant, thank you. So to follow on with that, have you tried to shorten the pacing protocol for functionally maturing cells?
Okay, I will take that as well. So I know there are several reports using shorter pacing process than our protocol. I think that would be fantastic if you could have matured cardiomyocytes after 2 days electrical pacing. However, you must be very careful about the pacing power. So in our hand, we observe the cell stress and actually decrease cell variability when we reduced the pacing process to 2 to 3 days by increasing pacing power. So from a practical point of view, I know it will be very helpful if we could make the pacing protocol shorter than 2 weeks. So we are actually currently working on the new maturation protocol. Hopefully, we will be able to provide an alternative solution which can functional mature cells in a week, but with minimum stress to the cells. Thank you.
Thank you for that answer. The next question is a compound one, but it asks, what is the level of purity in your iPSC-Cardiomyocyte population before starting maturation, and what is the level of cell death when recovering the iPSC-Cardiomyocytes for downstream applications after maturation?
Yes, the ICL-cardiomyocytes and the ICL-cardiomyocytes squared are greater than 95% pure cardiomyocytes based on cardiac troponin T expression. And the second part of that question is really as far as the cell death. We observed negligible cell loss. As Xiaoyu mentioned, the cell index did not change, and we did not see any significant loss of the cells.
Thank you. And suppose this work for iPSC-Cardiomyocyte monolayers?
So I will answer that question. So for this platform, actually, we prefer the cell monolayer. So all this experiment we performed just use the cell monolayer. So for the single cell stimulation, we haven't tried that yet. Thank you.
Thank you. Question from Marcus says, do you replate paste cells for other readouts like calcium flux, voltage dye measurements, etc.?
Yeah, I'll take this one. So all the experiments that I showed were done in the new ASEA plates that have a viewing window, so one does not need to dissociate and replate the cells for their experiments. The cells were pasted on the system and then moved to another system without replating them.
Thank you, TK. According to Comprehensive In Vitro Proarrhythmia Assay report, ZIPA report, it seems that immature iPSC cardiomyocytes can well predict proarrhythmia liability of drugs. Is it necessary to mature these cardiomyocytes before compound test?
Okay, I will take this question. So we definitely realize the current model used in the CEPA has very good predictivity and it's fit for purpose. However, as we have shown that there are a few compounds actually were miscategorized during the CEPA study. So according to actually based on our gene expression data, we already did some gene expression. We observed expression level increased actually in sodium channel beta isoform, which is the key regulator for the late sodium current. So as we know, the standard human iPSC-derived cardiomyocytes lack of late sodium current. So this result may explain why the long-term pacing improved the cell response to the safe compound rhinologine, which used in the CEPA study. So in addition, the expression of the gene for IK1 current was also significantly increased by the long-term pacing. So taking all this together, I believe that improving cell maturation before compound addition will increase the predictivity for drug. Thank you.
Thank you. I've got a question here about the high risk TDP causing compound that was misclassified. Would you have a solution for this?
Yep, this is TK. So one of a recent group has shown that if one calculates, I believe it's the free concentration of the compound, they are able to identify Beprudil. So it looks like with that combination of doing that and doing the pacing, one can correctly identify the majority of the CEPA compounds.
Thank you. How long can cells remain mature after stopping stimulation? Do they go back to an immature state?
Actually, we tested this. I think this maturation, we call this functional maturation, is transient. So it can last about 7 to 12 hours after you stop the long-term pacing. So this is what we have been observed.
Thank you. How do you interpret the reduction in beating amplitude after long-term pacing compared to non-paced cells, which was, I believe, shown in TK's data?
Okay, also this is actually based on our gene expression level. We detected the L-type calcium channel expression is actually reduced after long-term pacing. I think this is actually a good sign because there is a paper actually showed that this human iPSC-derived cardiomyocytes has higher expression level of this L-type calcium channel compared to the adult cardiomyocytes. I believe this reduction in the beating amplitude is related to the reduction of this L-type calcium channel expression. Thank you.
Thank you for that. I've got a question on the L-type calcium channel blocker. So how do you interpret beating rate increase after L-type calcium channel blockers, nifedipine addition? Do you see any difference between paced and non-paced cells after this addition?
Yeah, so this is TK. I'll take this one. The response for the L-type calcium channel blockers was the same in the paced and unpaced groups. There was recently a publication from Merck that showed that if you modulate the extracellular potassium channels or the concentration of potassium, one can make the cells have a slower beat rate with the addition of calcium channel blockers. So similar to this overall presentation, one can do different things to bring out different phenotypes depending on what the researcher is looking for.
Thank you for that, TK. And has there been a noticeable metabolic change in post-pacing cardiomyocytes?
So I will take that question. So for this part, we actually haven't really done any work on this test. So, but as you know, ASEA Biosciences right now is part of Agilent. So Agilent does have a very good kind of a platform to test metabolic. So we are going to this is actually on our agenda. We are going to collaborate with our team from Seahorse. We are going to test the metabolic change after long-term pacing. This is an ongoing project. Thank you.
Thank you. I've got another question about the monolayers, actually. So are the iPSC-Cardiomyocyte monolayers a necessity, or is it realistic to pace cardiac aggregates? And how about the ECM? Are there any ECM coatings that work well, or do they need a specific one?
So I can take a stab at this one. The only experience that we have with this system is plating the cells as a monolayer, and the ECM we used was fibronectin, and so we haven't really tried different formats.
Thank you. And I've got a question here. How do you use cardio ECR to analyze arrhythmia events?
So the cardio ECR instrument has two different readouts. So one is the impedance, which actually can evaluate contractility, and also the other readout is field potential signal. So when we evaluate arrhythmia, we can just use these field potential readouts to check these waveform, the field potential waveform change, if we can detect any EAD-like phenomena. On top of that, actually the impedance readouts, which reflect contractility, also can help us to evaluate arrhythmia. If the compounds cause arrhythmia, you definitely can see a rhythmic beating actually pattern showed on the impedance readout.
Thank you. How long do you have to wait after replating to perform other assays?
So I think for this one is actually cell-specific. So for example, for the iCell-Cardiomyocyte from CDI, for these cells, in order to perform assay, if you don't want to do the pacing, definitely you can do the compound addition. Normally it takes a week after seeding. For other cell lines you purchase, for example, from Ncardia, their cells also requires about 6 to 10 days in order to perform the assay, in order to add the compounds. So for the system we are using here actually have some cell status QC. If some cell line you don't know when you can add compounds, you can use our cell status QC protocol to evaluate the cell performance before you add compounds. Basically, you have to check whether the cells can generate robust beating and field potential signal, and whether those signals are very consistent. If the cell meets this criteria, that indicates the cell is actually ready for the assay. So it's actually, in one sentence, is that it really depends what cell lines you're using for your assay.
Thank you for that. Sorry, thank you. We've got quite a few questions coming in. We've got time for a couple more. So this one is, what's the difference between the iCell-Cardiomyocytes and iCell-Cardiomyocytes 2 used in TK's data, if you want to take that, TK?
Yeah, I can take that one. So the iCell-Cardiomyocyte squared, for that cell type, we tweaked the differentiation a little bit to allow the cells to be able to use sooner. So for those cells, one can use them as early as day 7 or day 4 to day 7. For the original iCell-Cardiomyocytes, you need to wait approximately 14 days. In the experiment that I showed, the iCell-Cardiomyocyte squared and the iCell-Cardiomyocytes are also from 2 different donors. So there's going to be donor differences between those cell types, but the differentiation process is just tweaked at the end.
Thank everyone as well for their questions. I think that's pretty much all the time that we have for today. I'd like to thank both the speakers for today's informative discussion and presentation, and thank you for going through quite a lot of questions we've had today. Thank you for everyone joining us online and for your interesting questions, and I hope you found this a worthwhile session. If we do have any more questions or you want further information, please feel free to email me at editor@selectscience.net. Excuse me, and I will follow up with your questions for both our speakers. Remember, you can also download related resources in the tab at the bottom left of your screen, which will include a certificate of attendance. If you'd like to listen again to today's webinar or invite a friend to listen, it'll be available to watch on demand in a few days. Goodbye, and thank you once again for joining us.